ci sono Marco? No, no, sto parlando. 1, 2, 3, prova, 1, 2, 3, prova, sì.
Prova. 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 No, è acceso. È arrivato? Siamo in diretta. Sono uguali il microfono.
Allora, poi si sente? Ok. Sì. Poi ascolta, quando ho fatto qua, te lo appoggio qua dietro e questo te lo metti tu qui.
Madonna, che mi siete
Va bene? A dopo. Ora lo devo tenere io.
studenti di master, per cui un po' di zoologia.
Di certo, laurea magistrale e triennale.
Allora, qua sono un misto di studenti di dottorato,
Mamma.
laurea magistrale primo e secondo anno e studenti della triennale del terzo anno, però seguono corsi diversi che prevedono tutte attività di seminario.
Io spero che l'aver tentato l'obiettivo cioè nel senso sulle conoscenze di alcune cose vuole dopo scontate.
No, ma dalle per scontato.
I seminari che vengono fatti in inglese di solito hanno come target o gli studenti di magistrale o di dottorato, e gli studenti della triennale vengono ma si devono adattare.
Si arrangia.
Prima faccio un annuncio. Ragazzi, scusate, allora oggi faremo un leggero cambiamento rispetto alla volta scorsa, cioè il question time siete liberi di farlo sia in italiano che in inglese. Siccome ci sono due studenti internazionali, loro ovviamente faranno, se loro fanno domande in inglese, la dottoressa Dani risponderà in inglese, ok? Però siete liberi di fare domande in italiano e la risposta sarà in italiano, ok? Oppure, se vi va bene, potremmo tradurre la domanda in inglese e poi rispondere in inglese, va bene? Ok, Francesca? Allora... Ok, si parte? Hello, everybody. Good afternoon, and thanks again for being here to our third seminar of our seminar series on dialogues on animal behavior. So tonight, this afternoon, we have Dr. Francesca Dani from the University of Florence, and I'm here again to introduce her. Dr. Dani and I, actually, we know each other since too long, and she got her degree at the University of Florence and her Master of Science equivalent on castal differentiation in social wasps. And then following that kind of taxon, she started working on chemical communication during her PhD again at the University of Florence with Professor Turilazzi, and on chemical communication on social wasps. After that, she got a very important award and scholarship, the Marie Curie postdoc fellowship, and she went to UK to the Keele, what's the pronunciation? Keele University in UK, where she kept working on chemical ecology. So she actually has become one of the most relevant scientists in chemical communication and chemical ecologist in insect. She then, after the period after the Marie Curie fellowship, she came back and she started working on the Mass Spectrometry Center at the University of Florence as a research associate. And then she was recruited, finally, and luckily, at the University of Florence at the Department of Biology, where she is now a researcher. So I leave you the podium.
Mamma mia, come siete tecnici, tecnologici, aiuto dell'altro. Ok. Acceso? No? Okay. So, can you hear me? Yes. Mi sentite? No. I don't know. I don't know what I have to do. Okay. I think that one is probably better. So this one and that one. Okay. Thanks. Good afternoon to everybody, and thanks to... No? No? Doesn't work. So it's me then. So just, it's me. Okay, can you hear? Okay. So thanks to be here, and thanks to Alessandro Massolo for inviting me. So the seminar of this afternoon will be about social insects, about chemical communication in social insects. And you will be listening a lot about social wasps, and in particular about paper wasps. Paper wasps have been a subject of researches in Florence since the 50s, and before they have been studied here in Pisa by a very eminent scientist called Leopardi, which was born in Pisa, has worked in Pisa for several years. And these are very fascinating animals. They have been becoming a model in eusocial insects. And before starting to talk about communication, I think it's worth spending a few words about what sociality, and in particular eusociality, means. So eusociality requires two conditions. The first one is that there must be cooperative brood care, including care of offspring from other individuals, which means that females, normally we are talking about females, care of other females' brood. The second condition is that there must be an overlapping of generation between adult individuals inside the colony. And the third one is that there must be a division of labor. A division of labor means, in this case, that there are reproductive individuals and non-reproductive individuals. But this latter condition can be reached in two ways, in two different ways. In the case of Polistes wasps, which are these nice animals here that everybody knows, there are females which are not very different from each other, and you have females which lay eggs and the other which don't lay eggs, but they may switch. So the one which don't lay eggs may suddenly become egg layers. So they may become queens, let's put easily like that. Then you have other species, like, for example, the hornets, in which you have a queen, and the queen is much bigger than the workers, and workers don't lay eggs, or they lay eggs very, very rarely, under very peculiar circumstances. Otherwise, they really don't lay eggs. And this condition is found across social wasps, but also across bees. Also in social bees, you find these two conditions. While you don't find anything like that in ants. So more in ants, this is the condition. So you have a caste of queen and a caste of workers. There are a few exceptions in a small family of ants, but we won't talk about that. So where do we find eusociality in insects? So as all you know, we found eusociality among termites. They are not called Isoptera anymore, but doesn't matter. And then we find eusociality among the hymenopterans. And so we found social species among wasps. Not all wasps are social, but many are social. And similarly, we find sociality among bees, but again, not all bees are social, while all ants are social. So we don't have non-social ants. And then we have other different situations, other orders where we find eusociality. So we have social aphids. Aphids may live in very small nests, let's put like that, so inside galls. And inside galls, they develop in clonal societies where there is one individual laying eggs and the other becoming soldiers. So they defend these galls. And the similar situation is also found in social thrips. Thrips are tiny animals. They live in some silk tunnel. They make these tunnels by themselves. And we found some fertile individuals and others which are soldiers, and they have specialized organs. Okay, here. And they use these legs to defend the colony. And as you all know, we also have some, but just a few species of social mammals. Someone says that we also are social, so eusocial, in the sense that if we consider women, all women, when they are not fertile, could be considered as a sterile caste. And so we could meet the third condition of eusociality. But I think this is more a joke than any other than true, but that there is a paper about that. Okay, so eusociality is a great thing, and it's such a great thing that Schmidt and Samari, in their book of Major Transition in Evolution, have inserted eusociality as one of the major transitions in evolution. Okay, so these are the major transitions, and eusociality comes just before the origin of human languages and culture. So I think it's relevant to think about what eusociality means. And of course, if you have social species, you have emergent properties. It means that in social species, if you consider the colony can perform tasks that single animals cannot perform, and you cannot see the colony as a sum of the single task of the single individuals, because they can communicate and they can organize behavior and do very remarkable things. And as an example, I would like to consider this species here, this ant. We have been studying this ant recently. This is Ecophila smaragdina. It's also called the green tree ant. It's found in Australia, in the southeast of Asia. And this is a peculiar species because the nests are on the trees, not in the ground or not under the branches, but they are made with leaves. And to make this nest, the workers do things like that. So they form chains. They form chains in which each ant keeps with the mandibles another ant, and then they pull the leaves close to each other, close, and then finally they use the larvae. They use the larvae to stitch together the leaves. So the larvae emits some silky stuff from the mouth, and this is how these nests are made, and they can be very big nests actually. So this is of course a good example, I think, for emergent properties. And when we think, I don't know why it doesn't work. Okay, when we think of organization, of social organization in colonies, of course, every one of us think about the honeybee dance. The honeybee dance is a very interesting way of communicating information. So by using this dance, the foragers, when they come back from a flight to flowers, communicate to the rest of the colony where they have found the food, and if the food is good enough to leave the nest and search for this food and go into this food source. So this is a very interesting form of communication, but actually in social insects this is quite unique. I mean, there are not other social insects which communicate like that. We find this form of communication only inside the genus Apis. We don't find it outside of the genus Apis. The most common channel of communication in social insects is chemical communication. So they mostly communicate by chemicals, not only by chemicals, but mostly by chemicals. And if you dissect an ant, or if you dissect a wasp or a bee, what you can easily observe is that they have so many exocrine glands. So they have exocrine glands everywhere. So in the head of the ants, you find big exocrine glands, which are called hypopharyngeal, postpharyngeal glands. They are huge. They produce many chemicals, and you find mandibular glands, which often produce alarm pheromones. And then you have glands on the legs, and they produce trail pheromones, and trail pheromones are also produced in the abdomen from other glands. So they are really a chemical factory. And when we talk about pheromones in terrestrial animals, the chemistry, the chemical nature of these compounds can be very diverse, but there are a few rules. So if you want volatile pheromones, the molecular weight has to be below 300 Dalton. But if you want contact pheromones, because you may want contact pheromones, then the molecular weight is higher and can be up to 600 or even more Dalton. And as I said, the chemistry can be very diverse. I mean, there is no prediction. You cannot tell the function of a compound on the basis of its structure. You cannot say, because you are an aldehyde, this is an aldehyde, this is, I don't know, an alarm pheromone. It doesn't work like that. You have to test all these compounds. And this is, as an example, I got this picture, which refers to the alarm substances in ants. There are not all the compounds which have been reported, there are just a few. But I think it's a good example. So you find ketones, you find alcohols, you find esters, you find aldehydes, you find pyrazines, you find sulfides, you find terpenes, you find hydrocarbons, so you find all sorts of compounds. The only point is that if you want an alarm pheromone, it has to be pretty volatile. Okay, so that's the only rule. So how do you analyze these compounds? How do you identify these compounds? This is quite easily, I mean, quite, not sometimes very difficult actually, done using gas chromatography coupled to mass spectrometry. So you first, because you often have complex mixture inside the glands, you dissect the glands, you separate the compounds using chromatographic method, and then these compounds are sent to the mass spectrometers, and then you get a mass spectrum, and the mass spectrum is a sort of fingerprint of your molecule, and hopefully you may find this mass spectrum in a database and you can identify the compound. But of course, then you need the chemistry to synthesize the compound to verify if your identification was correct. So this is just the first phase of the analysis. And again, using the honeybees as a model, we can talk of the most famous pheromone of the honeybee, which is the honeybee, the mandibular gland pheromone. And here you see the queen, and the queen you see is surrounded by workers. This behavior is called queen retinue. So they continuously follow the queen, and they touch the workers. The workers touch the queen all the time, and then they touch other workers. So this is probably a way to let know to everybody in the hive that the queen is alive and is still fertile. Okay, this is what probably it means. But these pheromones, the pheromone emitted by the mandibular glands of the queen has many different functions and diverse functions. So it regulates different aspects of the social life. So it tells workers that they have to keep rearing worker larvae, because the caste determination is due to the way in which larvae are fed in the first stages of their life. So if the queen emits these compounds, then it is sure that the queen is okay. Or if there is not enough of this pheromone, then the workers may start to build queen cells and to rear queens. And also it has a different effect. It reduces fertility in the workers. And here you see the ovaries of the queens. They are very big, full of eggs, while here you see the normal ovaries of workers. Sometimes you even don't find them. If you dissect workers, you often don't find the ovaries at all. But sometimes they are a little bit developed or quite developed, like in that case. This means that these workers can lay eggs, and they only lay male eggs. So the individual will be a male because they are parthenogenetic. And the same pheromone also acts as a sex pheromone. So it attracts mates from very far away, and the queens only mate at the beginning of her life, and they mate with several males. They can mate 20 times, and they store the sperms for the rest of their life in the spermatheca, and that's why they need all these sperms. And so these pheromones may have two actions. It can work as what is called releaser, so it releases behaviors immediately, like release attraction towards males, or it may have a primer effect, so it doesn't really release a behavior immediately, but it may reduce the fertility of the workers. If this compound is not present, then workers start to lay eggs. It also has an effect during swarming, because during swarming, the workers search for the queen, so they follow the queen. If the queen moves, then the workers follow her over a flight. But something that I like to make clear is that if we talk about queen pheromones, the idea is that the queen decides everything, controls everything in the colony, which is true, but not that true, actually, in the sense that in some contexts it looks like that, but in other contexts it's not like that. These are the compounds. We don't bother about the compounds. So this is a swarm, honeybees, and they may be searching, or certainly they are searching for a new place where to build a nest. And they have to make a decision. The decision has to be taken very quickly because they can only be out of a nest for 1 or 2 days, no more, because they don't have honey. And so they have to decide. And how do they decide where to nest? It's not the queen which decides here, but are the workers. So the most expert workers, which have been scouts or they have been foragers, go around and search for a new place, and then they come back to the swarm and they perform the dance, as we have seen before, and they try to recruit to the new place other workers, and the new workers go and they inspect the place, and if they like, they come back and again they dance and they recruit other workers. And so at the end, they finally the place is chosen on the basis of the number of dances which have pointed to one particular place. So I think this is a very interesting way of thinking of colonies. Colonies can be seen sometimes as very centralized structure, but sometimes they are not. They are not at all. It's can be very different organization. They also use chemicals to bring the whole swarm to the place. But in this case, this is again based on mostly on the dance, on this dance behavior. And now we come to a subject which has been my subject of research for several years, much less in the recent year, which is nestmate recognition. So if you look at social insects, you have to think that social insects have a very special ability, which is they are able to recognize the nestmates from alien individuals. So they can tell which one belong to which animals belong to their colonies and which one don't. And this is a very important feature of sociality, because if you think of a nest of honeybees or of a nest of ants or of a nest of wasps, these are very rich in food. So they often contain food which is collected outside and stored inside the nest, but the immature brood is itself food. So if someone can enter the colony and eat the mature brood, it finds plenty of food. And so they tend to defend their colonies against aliens individuals. Now, this is not that clear in the honeybees. The honeybees, of course, is a farm species, so, and often in case like that, you may have workers which enter a different colony, but this is a quite peculiar situation. I mean, in no farm in animals, it doesn't happen. But but if you sit in front of a hive, of course, you have to have the proper clothes to do that. But if you sit in front of the hive and you look at the work at the bees, you may see that there are animals which are in front of the hive, and they behave like guards. So they are guards. For a short period of their life, they behave like guards. And so when someone arrives on the nest, they interact with these bees, and they test them. So they touch them with the antenna, and they probably, in doing this with this behavior, can understand if these animals belong to the colony or not. And if they don't, they bite, they fight, they try to bring them away from the nest. It's very interesting and somehow funny to see how much energy they put in this behavior. And so how do they recognize each other? So we know that in social insects, the recognition pheromones are lipids which are found on the cuticle. So these lipids are have a different function. I mean, they had as first function to decrease water loss, to make the cuticle waterproof. But the second function has become a pheromonal function. So this was well, a good thing. I mean, if you okay, if you look at other arthropods, you don't have cuticular lipids. If you get ticks, mites, they don't have cuticular lipids, or they have very little. So while in insect, you see that they have developed a final function system, and at the same time, they have enriched their release of pheromones. So there has been a parallel evolution of the emitting system and of the perception system. And if you analyze this compound by GCMS, and then you do statistics on the composition of this data, you find that animals which belong to the same colony are very close for their cuticular lipid composition, while between colonies there are more difference. So this has been tested in so many species, which now we can say for sure that these are pheromones, these are recognition pheromones. But so far, I mean, if you consider just this, you may say, okay, well, they are more similar, but it's not really demonstrated that these compounds are real pheromones. But you may do something different. For example, you can change the profile of the cuticle by adding some compounds, which is something we have done ages ago on the honeybees. So we added some compounds which are normally present on the cuticle. They are not, I mean, these were synthetic compounds, but are the very same found on the cuticle. And so we implemented the concentration of these compounds. And then we put back the animals in the hive, and we check what guards would do. And what we observed was that, so controlled treatment had no effect, but other compounds do have an effect. For example, this is an alkene, an unsaturated hydrocarbon, increase the aggression by the guards. And similarly, these other compounds also increases aggressiveness, while other compounds don't. So it means that some compounds are more meaningful, others are not meaningful at all, perhaps, or less meaningful in this particular circumstance. But I said social insects can recognize nestmates. It's true, but they also make errors. They make recognition errors. And in the honeybees, this is probably the error rate is higher than in other species because, as I said, it's a farm species. But anyway, there are errors also in the other species. And we have used these errors to test the hypothesis that if cuticular hydrocarbons are really recognition pheromones, then the profile of when there was a mistake, when there was an error, these errors could be explained on the basis of the profile of the cuticular hydrocarbons. This was our hypothesis. So we had 3 colonies, and you see these are nestmates. In this case, we put nestmates on the colony. We knew because we have genotype of the individual, so we knew for sure that these were nestmates. And if you see when you have nestmates, most nestmates are accepted inside the colony, but a few of them are not. And on the other side, if you put non-nestmate close to the colony, then most of them are rejected from the colony, but some are accepted. So, of course, these are errors, and these are errors. These are recognition errors. So our hypothesis was that there should have been a correlation between the errors and the profile of the animals. And what we checked was how in case of error, the profile of the erroneously accepted or erroneously rejected individual was similar or dissimilar from the profile of the colony. And we were quite right, because if you look at the accepted individuals, these are the non-nest mates, so the alien individuals, and their profile is very similar to the one of the nest mates. So for any reason which we don't know, they were very close to the colony profile. It makes sense. And here it's a bit more variable, but these are the rejected individuals, and here are the non-nest mates, which are quite different from the profile of the colony we were testing in the experiment. And the nest mates which were rejected, as you see, they were very variable in their profile. So you have to compare this with this. So I think this is a good way to test the hypothesis that these are really pheromones. Okay, and finally, and I think this is the most interesting part of the story, we talk about social parasites. Social insects have several social parasites. As I said, nests and colonies are stuffed with food, so there are parasites trying to exploit this food or the nest and use them in a non-social way, let's put like that. And this is a parasite of Polistes dominola, which is the species you have seen at the beginning of the talk. This species is called Polistes sulcifer. We have 3 social parasites for Polistes wasps, and fortunately enough, they are all found around the Mediterranean Sea. They don't have anywhere else, so which make us very happy because we have worked with this wasp for a long time. And as you see, they are pretty different from the host species. They have a very big head, very strong mandibles, and they are quite nasty. So when they enter the nest, the host nest, when the nest is relatively small, they have, you see, there is some immature brood or just a few workers, and they fight with the queen. The fights can be very, very tough, and at least in the lab, most host queen are killed during these fights. We don't know for sure in nature, it's difficult to tell, but it occurs that you do find wasps under the nest which are dead, so we assume that it could have been the parasite. The parasite has no workers, so it uses the workers. They cannot build a nest and has no workers, so they exploit the workers by the host queen to rear its own brood. So these poor workers go away and they bring food, they build the nest, and they work very hard in order to breed the parasite brood. And this is very, weird, because we have just said that social insects can recognize each other and they can recognize conspecific, while here we see that they seem to be not able to recognize or to fight against the parasite. This is the parasite brood emerging from the nest. And ages ago, we have studied what happens with the cuticular hydrocarbons of this parasite. And if you get the parasite before they enter the host nest, they have a quite simple profile of cuticular hydrocarbons. It's very simple for being a social insect. Social insects normally have many more compounds. And this is the host at the same time. And if after 3 days, you find that the parasite has a profile which is very similar to the profile of the host. So they copy the profile of the host. There are a few compounds that don't seem to be completely able to copy the profile, but certainly they become much closer to the profile of the host after a few days. We don't know how it works. The most economical hypothesis is that the parasites get the hydrocarbons from the nest. The nest is also covered with hydrocarbons because these make the nest waterproof. And so there is a behavior in which the parasite stay in close contact with the nest. So probably there is a way to acquire these compounds from the nest. Someone thinks there could be a biosynthetic switch, but it has not been proven. So we don't know yet. This is not an exception. This happens in most parasites. We have studied this thing in parasite of ants, and it's always like that. I mean, it's a very common thing. Okay, and this is just the first part of the talk. The second one is much shorter, it has less to do with behavior, and is the other side of chemical communication. So far we have talking about pheromones, but now we talk about how pheromones are perceived. So from one side you have to emit pheromones, from the other side you have to smell pheromones. And this part of the story has started here in Pisa in a very interesting collaboration which started in 2005 with Professor Paolo Pelosi, which was a professor here in Pisa at the School of Agriculture. And he is a big expert in chemical communication and in perception. And we were lucky enough to be involved in a common project, and since then we have worked in collaboration on several projects. I have learned a lot from him, so it was really a very interesting collaboration to see things from the other side, so how odors are perceived. And so odor in insects are perceived with antennae, or mostly with antennae, not only antennae, but mostly with antennae. And as you know from your courses, the antennae are covered with some chitinous structures which are called sensilla, and in this case chemosensilla. And these chemosensilla, you see, have pores in the walls. And through these pores, odorants can enter inside the sensillum and are perceived. And this is the start of the olfaction. And I won't talk about how then the odors is processed, because I'm not expert enough to talk about that. So what we have been studying with Paolo Pelosi are proteins which are present in high concentration in the sensilla lymph. So here you have the neurons, the odorant receptor neurons, and here you have the dendrites, and the dendrites are in contact with the hemolymph, and they are in contact with these proteins. And And this is a funny cartoon made by Paolo Pelosi several years ago. Sometimes I use, sometimes I don't. So these are the soluble proteins I was talking about. These are the odorants, and these are the receptors on the membrane. And very simply, this is what happens. You can imagine that these proteins have an hydrophobic pocket, and inside the hydrophobic pocket they can get odorants, and of course the affinity depends on the shape of the odorants and on the shape of the pocket. And finally, they can release the odorant close to the receptors. And well, probably it's not that simple, but I think it's good to explain the whole story. And there are two families of these soluble proteins which have been studied by Paolo Pelosi, and the second one has been identified by him. So it was the first one to identify these proteins. The two families are very different in structure, pretty different, but both have this hydrophobic pocket inside. And they are called odorant-binding. The first is called odorant-binding protein, and the second one is called chemosensory protein. They differ in the number of cysteines and the way they form disulfur bridges, but we don't care much about that now. So what we have been asking ourselves was when have the genes for this protein appeared during evolution. As I said before, insects seems to be very good at olfaction, so they have a very remarkable sense of olfaction. But if you look at other arthropods, terrestrial arthropods, we really don't know much about olfaction. We know little, I would say. What we know is that odorant-binding proteins appear in hexapoda, so you don't find them before. They have never been reported, and they seem to appear together with the odorant receptors. So there is both family appears in hexapoda. And for chemosensory proteins, things seems to be different because there are a few genes. The size here refers to the number of genes. So we have a few genes of chemosensory proteins even in other groups of arthropods. So we have a few, for example, in ticks. But then again, it seems that the number increases in insects. So again, in insects, there is something going on, so about olfaction. And then recently we have described a protein proteins which are very well known in the animal kingdom. These proteins are called Niemann-Pick C type 2 proteins, and these proteins are carriers of lipids. So they have this general function of carrying lipids. But at the beginning, we started to identify this protein in the antenna of the honeybees in huge amount. There was a huge amount of this protein. And they are called, the name in the database was ejaculatory protein. And we were, why in the antenna? So we couldn't make a sense out of that. And anytime I would ask Paolo about that, I would say, why do we have this large amount, the large amount of this protein? I would say, don't bother about that. Who cares about that? And then finally, there was a paper on hand saying that they also found this protein in large amount in the antenna, and they thought it could be an odorant binding protein. And so we said, okay, this makes sense. And we started to consider this family of proteins more seriously after, 8 years, probably. And so these proteins are found also in other arthropods. So they may have the same function, or they may have acquired a different function than in insects. But this doesn't tell us if these proteins are found in the antenna or not. We are talking about the number of genes in the genome, not about the expression in the antenna. So we have largely used proteomics to study the expression of these proteins in the different part of insect bodies. And proteomics is, it's simple and it's difficult at the same time. So you can either run a 2D gel, which we have done in collaboration with Felicioli, with Antonio Felicioli here in Pisa, and I have analyzed by mass spectrometry the spots. Or you may skip the gels and you can prepare a protein extract and then you digest the whole extract all together and you inject that in an LCMS system and then you get spectra and of the peptides you have in your mixer and you identify the proteins using databases and you also can make a relative quantification of the different proteins in your samples or between different samples. So it's very easy to be done. And by using this way, we have this method, we have studied these soluble proteins in different animals and different insects, and other people have done using different methods, not proteomics or also proteomics. And for example, we have studied the expression of these olfactory soluble proteins in the mandibular glands of the queen, and we have found some differences between castes in the mandibular gland. And similarly, these proteins have also been reported for several exocrine glands. So these proteins are not only found in the antenna, they are found in exocrine glands. And the idea is that they may be carriers of pheromones. So in this case, they may be carriers toward the outside of the body. So they may serve to release in a controlled manner how pheromones are released to the outside. And so since then, they have been found in several different organs, and especially they have found in male reproductive organs. We don't know exactly why, but they are in large quantities. And these are a few things about the very recent work we have been doing, still using a proteomic method. And here we have studied the expression of these proteins in castes in the honeybees. Here we are comparing workers and queens. And what we have found is that there are some proteins which are more expressed in the queens, which are this, and some which are more expressed in the workers. Actually, we cannot make a sense of that. We really don't know why. I mean, does it has to do with their behavior? For example, one could think that foragers may need different carriers because they have to smell different compounds with respect to the queens which live inside the nest. So this is the hypothesis. This was not really a novelty because we knew that from other projects which have not used proteomics. And we have tried to see if we also could find differences between tasks. So if you get workers change their task during their life. There are nurses at the beginning of their life, and then they become guards. Not all of them become guards, but some of them become guards. And then they become foragers, and they die as foragers. And so the idea was trying to see if we could find differences between tasks. These have never been reported before, such differences. And we found some difference, not so many differences as in the comparison between workers and queens, but we could see, for example, that this protein here is higher in foragers with respect to nurses, while others are like this is higher in nurses with respect to foragers. This is a log2 scale, so it doesn't seem so different, but it is. If you have a difference of 1, it means that it's the double. So, which is a relevant. This is a relevant difference. But of course, I mean, we should. What should we do next? We should have this protein made recombinant and then test these proteins for their affinity towards a variety of compounds of environmental compound, volatiles or of volatiles which can be found inside the nest. So it's just the beginning of a long story. And very finally, I'm leaving you with this animal. This is varroa, the varroa mite. It's a very nice animal, very nasty and very nice. And as you may know, this mite is specialized on bees, on the honeybees. Has a very interesting story, but I won't talk about that. So I want to show you this. Why not? Okay, this very short video, and I mean, okay, that's in German. Sorry for that. Okay, you. So anyway, the story is that they don't, of course, they don't have antennae, but they seem to use the first pair of legs as antennae. So they very rarely use the first pair of legs to walk. But they move around keeping this first pair of legs in the air, and they seem to smell something. This is quite similar in ticks. Ticks do the same. And you see, these are the legs. They don't seem legs, actually, they seem antennae. And so what we have done recently is trying to see if we could find olfactory soluble proteins in the organs which have been described as chemosensory organs in this animal. And the organs is not on the whole on the whole leg, first pair of legs, but it's just in this area here. This is called Haller organ. And other chemosensilla are found also on the mouth, on the chelicera. And what we have, there was, well, science is like that. I mean, just before we submitted our paper, there was a group of very clever scientists in Israel, and they made a very similar experiments, but they didn't use proteomics. They made RNA-seq experiments, and they described some proteins which they called OBP-like proteins. And they call like that because they also have 6 cysteines like the OBP, although the structure is remarkably different from the one of the insect OBPs. According to Paolo Pelosi, at least, I mean, he's the expert who says, no way, they are not that similar. And what we have done is we have quantified this protein in the legs, which was quite boring because dissecting these animals takes plenty of time. And we also use as a control sample the second pair of legs, and we check if this protein were only found in the olfactory organ or if they were more expressed in olfactory organs. And actually, we couldn't find proteins which fit with, which could be olfactory proteins and which are only found in olfactory organs. This is not the case. There doesn't seem to be such a specialization like for some OBPs in insects. But we found that this is the ratio between the first pair of legs, the forelegs, and the second pair of legs, or the mouth part and the second pair of legs. And we found that some proteins are more expressed where you find the olfactory organs, which is an interesting result. So our, let's say, competitors in Israel described this protein, but they said, no, these proteins are expressed everywhere in the body, while we do find a difference. So again, next step will be to characterize these proteins and see if they really are involved in olfaction. Of course, there must be something, because these parasites can smell the very same compounds that are smelled by the honeybees. So inside the hive, they can recognize male larvae from female larvae. They prefer male larvae because they have a longer time of development, which is better suited to the development of the parasite. And so they have to have a way to find the males inside the nest, which is, I mean, the nest, the hive is huge for varroa mites. If you think how small they are and how big is a nest, they have to have a way. But so far has not been described. And I think, yeah, this is finished. These are a few people the few people have mentioned. Most of you, or you may know some of them. This is Professor Leopardi. This is my supervisor and now colleague Stefano Turillazzi, my colleague Rita Cervo. We were much younger, all of us. And Paolo Pelosi, and this is Ima Iovinella. She has been Paolo's student and PhD student. Now she's working with us in Florence. And this is Antonio Felicioli. And I'll thank you for the attention.
Thanks a lot, Francesca, for this excellent seminar. So we open up for questions. Anche in italiano. One over there.
So, thank you for your very presentation. I was just wondering if, regarding the lipid composition of particular organisms, if the difference are concerning the some kind of lipid concentration or the average in, or the nature of lipids, so different planets.
Okay, so in most cases, when it has to do with nestmate recognition, it has to do with concentrations. But in other cases, it has been reported that these compounds also differ between fertile and non-fertile individuals. And in this case, just a few cases actually, the fertile individuals may have different compounds. Or if not really, so these compounds may either be absent or they may be in a very, very low amount in the workers. So it depends, but mostly it's just a difference in concentration. But the point is that all these compounds are very similar to each other, so we really don't know how they can perceive and discriminate between similar, between the profiles. So how can they tell apart compounds which are very similar each other? This is has been is becoming a very interesting subject of research for neurobiologists interested in perception. So it seems that social insects are becoming very popular for that. We never have thought of that before. I mean, they are neurobiologists, they are much smarter than we are as entomologists.
Any other question? We do have one. So you said these kind of proteins, I don't know which ones exactly, they are also in venom glands.
Venom releases plenty of compounds which have a pheromonal activity.
And so why is that exactly? Is there any hypothesis about that?
Well, there are alarm pheromones in the venom, so they may be involved in the circulation of these volatiles and in the release of these volatiles. We actually don't know. I mean, we know that there is a huge amount of some of these compounds in the venom of the honeybees and also in the wasps, in social wasps. But I mean, we don't know why.
So they are likely to be transmitted with the venom itself.
Well, probably yes.
So
it could be.
Could they play a role in not activating, but letting the venom pass through the target? I don't know.
And nobody ever thought of that, but I cannot say no. I don't know.
eusociality has been shown. Just to update my slides for my.
Yep. But maybe mine too. Mine too.
Because you mentioned what you say, trips.
Yes.
Is it a novel?
No, it's not actually novel. It's they were discovered in the 90s, and there is a book by Crespi which is called was about social insect outside hymenopterans. It also reported about some crustaceans which are considered to be eusocial. So I didn't mention crustacea because I think it was.
It's shown that there.
Yeah, it is shown.
There's more shrimp. What is it?
Sorry? Yeah, shrimps. Yes, yeah, it's shrimps. So it seems that eusociality has evolved several times. So if we consider it as a major transition, this transition has occurred several times. As an example, it has occurred at least twice in the social wasps. In social wasps, we have a nice family, which is of wasp subfamily, which are called Stenogastrina. They have been studied they are studied by Stefano Turellazzi, and they have tiny colonies made of just a few females, and it has recently been reported that the evolution of Stenogastrina is completely different from the other social wasps. So it means that eusociality has evolved at least twice in social wasps. So it seems that under some circumstances, eusociality is favored. Okay, that's it.
I have a question about the bees reproduction. So you said queen bee reproduces once in a lifetime, right?
No, it's not a reproduction. Mates just one. So it mates with several males, but just one.
So what happens if a queen runs out about all the sperm?
It doesn't happen. It doesn't happen. I mean, or it becomes sterile. But if you ask beekeepers, they remove the queens after 2 years normally. I mean, that's the standard rule. Or some beekeepers don't remove them at all. They think that workers can do better than beekeepers themselves. But if you if you don't remove the queen, they can live up to 5 years, and I don't know. Finally, they may perhaps become sterile, but or they probably become old. I mean, if you consider that workers live on average 45 days, and I mean the difference is unbelievable. I mean, you have 45 days against 5 years, so it's a fascinating point how using the same on the same genotype you may have such a different phenotype and such a different lifespan. It's amazing.
More questions? Oh, yeah, Anna.
I have a question on the signature of the nest male. So where does it come from? Because it's under genetic controls. You don't know. Yeah. Because you have several lines. Because there is a female and then several males. So within.
You mean inside the hives?
Yes.
Okay, inside the hives, it's a mess. It's a mess. Someone thought that because you have different patch lines inside the hive, then workers may recognize each other. They may decide which are the workers which are more related to themselves, and so they could decide to collaborate more with the close sisters and to collaborate less with the half sister. They are called half sister if there are different fathers. This is a fascinating idea, but it doesn't work like that. Nobody has ever demonstrated that it works like that. And I tested this hypothesis in the hornets and in Polistes wasps, where you don't have different partial lines, but you may have different match lines, so more queens which lay eggs in the same nest, although one queen lay more than the others. But you cannot tell apart the lines on the basis of the cuticular hydrocarbons. It doesn't work. I mean, nobody has ever demonstrated so far. And the behavior doesn't show that they are nepotistic in the behavior. So what is the base? I mean, there are enzymes which are involved in the production, the biosynthesis of hydrocarbons, but these enzymes have been studied in solitary species, but never in social species. For example, we know many things about flies, the domestic flies. We know about Drosophila. We know very well that if the flies change their fertility, the cuticular pattern changes, so they have more compounds. There is an increase in some compounds, but we don't know anything about the enzymes in social species. But even if there is a genetic basis, so I would expect that animals which live in the same colony are more or less related at a certain extent, and so they may have a similar pattern of expression of these compounds. But then we know from the parasites that these compounds can pass from an animal to another or can be acquired from the nest. So I think it's a very complicated thing where you have partly something which depends on the genetics, something which depends on the food, and something which depends on the environment. And of course, then there are other things like in the ants. As I have shown, you have these huge glands in the head, and these glands contain hydrocarbons, and there are drops of something which we call saliva, but we don't know for sure if it is saliva, which is passed from a worker to another. And it has been assumed that this could be a way to maintain sort of standard profile inside the colony by through these prophylactic behaviors. So it's very complicated. We really don't know how much is genetic and how much is environmental.
Sorry, sorry if I step in. We did something together, or at least I helped with the statistics of a paper 15 years ago, that we tried to relate genetic relatedness with the chemical.
Was it with me?
You wrote it, and I was one of the co-authors.
Yeah, that's the one. That's the one.
Yeah, okay.
That's the one on the matrix lines, on the patch lines. But we couldn't find such a difference. I mean, if we try to, if it is, it's not a strong difference anyway. It probably wouldn't be enough to tell apart different matrix lines or different patch lines.
We found some.
Well, it was very weak. I mean,
yeah,
it was very weak. Extremely weak. And the point also is that since we don't know which compounds are really important for recognition, the only thing we can do is do statistics on the data we got from the GCMS analysis. Because we don't know which compounds are important. If we knew that, we could work in a different fashion, but we don't know. So we have to include all these compounds in our analysis.
I have another question. Is the nest signature stable over time?
No, it's not.
Yeah. So probably is affected by environmental.
Yeah. But yes. But also, larvae have a different profile, and if they contribute with the hydrocarbons to the nest profile, then of course the profile changes.
There is a question over there.
In the wasp, there are alarm pheromones, yes, certainly. And also in the honeybees, there are alarm pheromones. This is a general rule. But in ants, some ant species have alarm pheromones in the mandibular glands. They produce from the mandibular glands.
Any more questions? Okay, so thanks a lot, Francesca.
Thanks for
avresti da firmare.